Hello everyone. This time I will introduce another form of cell death, apoptosis. The definition of apoptosis is a pathway of cell death that is induced by a tightly regulated suicide program in which cells destined to die by activating enzymes capable of degrading the cell's own nuclear DNA and nuclear and cytoplasmic proteins. The morphologic change and biochemistry of apoptosis can be divided into three steps. Firstly, cell shrink. The cytoplasm condensed and became eosinophilic. The chromatin is also condensed. Then the nuclear collapse and cell membrane start to bleb. Finally, the cell membrane wraps chromatin fragments and cytoplasmic organelles to form many apoptic bodies. These apoptic bodies will be cleared by phagocytes. This picture shows the squamous epithelium. Here is an apoptic cell. It is smaller and separated from surrounding normal cells. The nucleus is condensed and cytoplasm is eosinophilic. Observed by the transmission electron microscope, during apoptosis, the nucleus DNA of apoptotic cells broke into nucleosome-sized pieces and aggregate at the periphery of nucleus. Like here. Finally, the nucleus karyophysis. This picture shows us the differences between necrosis and apoptosis. During apoptosis, the membrane is intact. The cell shrinks and membrane blebs, finally form apoptotic bodies. Apoptosis would not cause adjacent inflammation. During the necrotic process, the cell swells, the membrane fractures, cellular contents leak out. At last, necrotic cell is digested by lysosome. This table shows the differences between necrosis and apoptosis. They are different in cell size, nucleus, plasma membrane, cellular contents, adjacent inflammation, and physiologic or pathologic roles. You can read it after class. Now, let's move to the significance of apoptosis. Apoptosis can eliminate cells that no longer needed in our body, maintain a steady number of cell population, also keep the damage under control. Apoptosis occurs in many situations. First one, the physiologic causes are the programmed destruction of cells during embryogenesis. Secondly, cell loss in proliferating cell populations. For example, apoptosis participant in renewal of intestinal epithelium. The third, death of cells that have served their useful purpose. For example, in the foci of inflammation, neutrophils will undergo apoptosis in the end of inflammation. The fourth, elimination of potential harmful self-reactive lymphocytes. And the fifth, cell death induced by cytotoxic T lymphocytes. The pathologic causes of apoptosis includes the first, elimination of cells with injury may be a better alternative than risking mutations in the damaged DNA, which may progress to malignant transformation. Secondly, accumulation of misfolded protein culminates in apoptotic death of cells. The third, cell injury in certain infection, particularly virus infection. The fourth, pathologic atrophy in parenchymal organs after duct obstruction, such as that occurs in kidney. Cellular aging. This part is for self-study. Now let's take a summary. We know when cells encounter severe stress or the persistent stimuli become deadly, cell death happened. It has two forms, the necrosis and apoptosis. We study the causes and pathological changes of both forms. Here is a question for you. What is the differences between necrosis and apoptosis?